Hello, this is Don Withers, and this video covers the electrical system of the heart. The electrical system, including the pacemaker and other structures, generates rhythmic contractions spontaneously without assistance from the nervous system. The heart pacemaker is part of an internal wiring made up of cells called nodal cells or autorhythmic cells, and these are what generate the electrical stimulus for contraction. The action potential is the result of the activity of these electrical cells. And this action potential generated from the pacemaker travels throughout the heart. And when it interacts with the cardiac myocytes or muscle fibers, it eventually causes them to contract. So in this video, we will look at the following. First, we'll describe the anatomy of the intrinsic conduction system, or the wiring that I mentioned before. What are the structures involved, and where are these structures located? Next, we'll look at how an electrical stimulus, the action potential, is generated, and how this action potential is spread throughout the heart. And then, lastly, we'll look at the mechanism by which the heart's pacemaker generates these rhythmic action potentials. These involve structures in the plasma membrane of the cells in the pacemaker and ion transporters that are involved in generating the electrical stimulus. This figure shows, on the left, shows the structures that are involved. And in this figure, the intrinsic conduction system, the electrical wiring, is tinted in yellow. These structures are all located within the myocardial layer of the heart, and they're embedded within and among the cardiac myocytes, and they're in close contact with those cells. There are 5 structures that are involved, and they're named the sinoatrial node, and this is the only part of the intrinsic conduction system that is off by itself, not directly connected to the other structures. The sinoatrial node, or SA node, is known as the main pacemaker of the heart. So this is where the action potential is generated and first starts spreading throughout the heart. The SA node is located in the upper or part of the right atrium, or often referred to as the roof of the right atrium. Next is a packet of cells called the AV node, and the AV node lies in the floor of the right atrium. This is connected to other structures in the intrinsic conduction system. From the AV node is a bundle of nodal cells that make a pathway through the interventricular septum. This is called the AV bundle, or atrioventricular bundle. The AV bundle branches within the septum to form 2 main branches, and those are called the bundle branches. Those extend to the apex of the heart, where next the structures run up the walls of the ventricles, and these extend also into the papillary muscles that protrude or project from the interior wall of the ventricles. These end in expanded paddle-like structures called the Purkinje fibers, and those radiate throughout the ventricular walls. All those structures are listed in the table on the right, and their location is indicated as well. Next, let's discuss how these structures, the intrinsic conduction system, made of nodal cells, works. First of all, a little bit about these cells. These cells, although they're embedded in the myocardial layer, are not contractile. The main job of these cells is to generate an electrical impulse and to spread that impulse throughout the heart. This is the main stimulator for generating contractions rhythmically and synchronously. So just some terminology that's involved here. Each contraction in the heart is triggered by an electrical impulse, and that's called an action potential. And when we talk about action potentials, we'll discuss 2 main parts that are involved. Part of an action potential, the first part is when cells depolarize, and that's followed by repolarization. This involves the distribution of ions inside and outside of the cell membrane of these cells, and we'll go into more detail about this in future slides. First of all, let's talk about how the impulse, the action potential, spreads among the structures that we discussed previously. Everything starts at the pacemaker, the SA node, and then the electrical events spread throughout the heart, through the other structures, and also through the myocytes. So let's look at this pathway first. In this figure, we can see that the action potential starts at the pacemaker of the heart, the SA node located in the roof of the right atrium. The electrical events that occur involve ions moving into and out of cells. And not only that, but between cells. Cells in the myocardium, both the nodal cells in the SA node and the muscle cells in the wall of the atria, are connected by passages through the membranes. And the ions that are involved in these electrical events can therefore pass from cell to cell and cause an action potential that starts in the SA node to spread through all connected cells in the walls of the atria. That includes both the right atrium, where the impulse was first generated, also to the left atrium through a pathway that's partially obscured by the vessels shown here. The structure that connects neighboring cells and allows ion movement are called gap junctions. You can think of these as simply pores allowing ions to move. And so when an action potential is generated in the cells of the SA node, it will move through ion movement through neighboring cells to all of the atrial muscle fibers in both the left and right atrium, and eventually to the cells of the atrioventricular or AV node. This figure is just showing how, and if this is the SA node, how an electrical event can be spread through gap junctions to neighboring muscle cells, and movement of charged particles is essentially a current. So that's what we have here, a current moving from the SA node to all connected muscle cells, and then eventually to all their neighbors, and to their neighbors, etc., until it spreads entirely throughout all of the atrial walls. The next event that happens is that the action potential through spread of ions through gap junctions reaches the AV node. Here, there's a delay in transmission or conduction through the rest of the intrinsic conduction system. The AV bundle would be next, and the bundle branches. The delay is due to the fact that the cells of the AV node are relatively small and they have very few gap junctions. So it's going to take a while for this electrical event, the action potential, to spread through the cells of the AV node and then be conducted on through the rest of the structures into the ventricles. An important point to make here is that this electrical event that starts in the atria at the SA node has only one pathway to get to the ventricles, and that's through the AV node and the rest of the intrinsic conduction system. The other pathways are blocked, not really shown on the slide, but the blockage is due to a thick bundle of fibers present at the border between the atria and the ventricles. Because of that thick bundle of fibers, essentially an electrical insulator, only one pathway is open for this electrical signal to reach the ventricles, and that's through the AV node and the rest of the intrinsic conduction system. Well, after the cells in the AV bundle pass on or conduct the action potential after this slight delay, it's going to enter into the next structures involved, the AV bundle, the bundle branches, eventually reaching the apex of the heart, where now the electrical stimulus, the signal, is spread to the Purkinje fibers running up the walls of the ventricles. The conduction, again, is through connection of all these cells that we've talked about in the SA node, the AV node, the rest of the intrinsic conduction system, the muscle fibers in the walls of the atria, and the muscle fibers in the walls of the ventricles. It's spread through gap junctions connecting all of those cells together. This figure just demonstrates that the conduction of the impulse to the ventricles happens by spreading very rapidly through the Purkinje fibers, which are large, and that allows the electrical impulse to be transferred to ventricular muscle fibers very quickly. So there's essentially synchronous stimulation of all of those fibers, and both in all sides of the ventricle walls and also in the papillary muscles. The end result of an action potential spreading through first the atria, and then after a pause through the ventricles, is for the muscle fibers to receive this electrical stimulus, and in response, they generate contractions. The action potential, when it reaches the muscle fibers, travels across the membrane of these fibers, it internalizes, and the internalization causes the release of calcium inside the cell. And then calcium is what causes the contraction, the actual contraction of the muscle. So, to summarize, the electrical events are the stimulus, those spread throughout the atria and the ventricles, and that those cause the mechanical event, the contraction, to occur. Okay, so far we've talked about the structure of the intrinsic conduction system, which generates and propagates or transmits an electrical impulse throughout the heart. Now let's turn to how the pacemaker, the cells in the SA node, generate rhythmic action potentials. And keep in mind that the pacemaker is solely responsible for causing heart contractions. It can do so independently of the nervous system. So let's turn to the nodal cells themselves and look at how they generate an action potential. And a few things that we need to know about these cells is that these cells have what's called a resting membrane potential, in which there's a net negative charge on the inside of these cells and a net positive charge on the outside. The difference in charge, or charge separation, is due to the fact that ions have an uneven distribution on the inside and outside of the membrane. There's generally an excess of cations, like sodium and calcium, outside the cell, and there's an excess of anions, protein ions mostly, on the inside of the cell. Also, keep in mind that there are ion concentration gradients. For sodium and calcium, the concentration of those ions are greater outside the cell. And if an ion channel for either of those were open, they would migrate by diffusion to the inside of the cell. Just the opposite is true for potassium ions. They have a higher concentration inside the cell, and when potassium ion channels are open, they migrate to the outside of the cell. So important in our discussion will be these concentration gradients for sodium, calcium, and potassium, as well as the ion channels that allow them to cross the membrane. These ion channels that are present in the plasma membrane of the nodal cells are voltage-gated channels. They open in response to changes in voltage across the membrane. So let's take a look at those, and then we can talk about how an action potential is generated. In this figure, we're looking at a representation of a nodal cell. This could be a cell in the SA node, and it has the resting membrane potential. That's what RMP means. And the value of this is indicated as minus 60 millivolts. The negative charge is just indicating that there's a net negative charge inside the cell relative to the outside of the cell, which is comparatively positively charged. This shows the concentration gradients for calcium and sodium, high concentration outside the cell, and for potassium, high concentration inside the cell. And at the bottom of the figure are three different ion channels. They're color-coded just to keep them straight. And these are going to be important for allowing ions to cross the membrane in response to some voltage change. So, for sodium and potassium, sodium and calcium, for sodium and calcium, if this is the cell, the plasma membrane represented here and here, this is the cytoplasm, cytosol here, and the interstitial fluid outside, sodium would tend to move inside the cell down its concentration gradient, as would calcium. Potassium, however, would move outside the cell down its concentration gradient. All right, so let's talk about the steps involved in generating an action potential. And these steps involve basically a series of opening of the ion channels that we just looked at. Each ion channel will open in response to the membrane reaching a particular value of membrane potential. One thing to keep in mind is that these cells in the SA node have an unstable resting membrane potential. Unlike all other cells in the body, the membrane potential, when they're at rest, tends to change without any stimulation. This is key to how these cells work to generate continuous rhythmic action potentials. We'll take a look at what this unstable resting potential means, but before we go to the next slide, just a term here. This unstable resting membrane potential is also called the pacemaker potential. Okay, here we go. These are the steps involved. For each action potential, it's a cycle in which there are 3 events. Each event happens in series 1, 2, and then 3. For each event, they're triggered by opening of an ion channel due to a particular membrane potential value being reached. In step 1, at minus 60 millivolts, the slow voltage-gated sodium channels open, allowing sodium to flow into the cell, or sodium influx. This is the pacemaker potential, which I mentioned before. As sodium flows in, the membrane potential value changes in the positive direction. When it reaches minus 40 millivolts, this triggers step 2. Step 2 is opening of fast voltage-gated calcium channels. And this allows a very rapid influx of calcium into the cell. This causes a very significant change in the membrane potential such that it reaches 0 millivolts. This triggers step number 3, in which the calcium channels close and fast voltage-gated potassium channels open. When potassium channels open, potassium flows out of the cell, and that brings the inside of the cell back to a more negative value. And so the membrane potential drops until it reaches minus 60 millivolts. Minus 60 millivolts is the stimulus to trigger step 1 and start the next cycle. These changes happen very rapidly, less than a second typically, and so then that's the results in the next round of action potential, which will eventually cause a contraction. Okay, we're almost at the end here. This slide represents what we just talked about on the previous slide and shows it in more kind of a graphical and as well as pictorial form. Here are the 3 steps, number 1, number 2, and number 3. And these are linked to the changes that are happening in membrane potential on this graph. So, step 1 starts with the membrane potential being at minus 60 millivolts. Right, in step 1, the sodium ion channels are open. Minus 60 millivolts causes them to open. That's the value. And when they open, sodium flows into the cell. Positively charged sodium ions flow in and cause the inside of the membrane to be less negative or to move in the positive direction. And that's what we see here. These are slow voltage-gated sodium channels, and the sodium doesn't flood in, it more dribbles in. And so there's a gradual change in the membrane potential from -60 upward towards 0. Here it reaches a critical value called the threshold value, which causes step 2. In step 2, the calcium channels open, and these allow calcium now to flood into the cell. And as a result, there's a very rapid depolarization of the membrane, movement in the positive direction. When it reaches 0 or slightly above 0 on the positive direction, that causes step 3. In step 3, calcium channels close and potassium channels open. Now potassium is going to leave the cell and positive charges will be exiting. This is going to cause the inside to become more negative once again. And that's what we see here in step 3. Now it goes from 0 in the negative direction, and it's eventually going to reach -60. -60 starts the cycle all over again. Potassium channels close and the sodium channels open, causing the next pacemaker potential to restart the cycle. Notice that this happens cyclically. 1 causes 2, causes 3, causes 1. And so this is constantly going to be happening over and over again. And this happens spontaneously without any stimulation from the nervous system. So the pacemaker is driving these electrical changes through these series of steps. Okay, that ends this video. In the next video, I'll talk about how these electrical events in the nodal cells are going to cause a similar but slightly different set of electrical events in the cardiac myocytes. And then we'll talk about how these electrical events can be monitored by electrocardiography. Okay, see you next time.